from virus nucleotides. From viruses have supplied our building blocks. So we're composed of blueprints, operating systems in the form of DNA, and our chromosomes have about 3 billion nucleotides, and onions have 15 billion. Each nucleotide is part of a sequence of hundreds, thousands, a million that together determine when, how, and which type of protein or enzyme gets made. And pretty much the rest of everything else is small stuff. I want to begin in the beginning. So in the beginning of the Earth's biosphere, molecules formed patterns as each one had specific bonding characteristics that worked best with another molecule. Molecular patterns cycled in pulses of scale or complexity driven by sun and darkness or tides or temperature gradients. Some absorbed more energy, maybe duplicated some residues which joined up and formed protective surfaces. Because molecules specifically bonded with other molecules, some could detach and reattach to new partners, but form the same molecular pattern. Patterns aggregated, they reproduced what they'd come from. Those better protected, able to absorb a little more energy, better able to exploit the cycles, they started to generate copies of themselves, and something like this is how life emerged. But below this threshold is a thick continuum of molecular activity. The notion of emergence is stuff rising from non-life into life. So there are patterns that are not sufficiently assembled to duplicate. They share bonds with each other too, and they have charge distributions that let them attach to the emergent patterns that can duplicate. Because after all, it's through these bonds that everything does, all assembly occurs. So some of these pieces could participate in the duplicating things, the bigger productions, attaching to the ends or the middle of the replicating groups, and their little reduced size let them bond to the larger unit, then get duplicated and then separate. So emergent life becomes this growing population along with all these interlopers naturally attracted to their molecules. Some joined maybe to the bigger ones, most maybe didn't even make it, but plenty were able to exploit it, hitchhiking and getting reproduced. So these diminutive parasites, which I'll call electron microscopic parasites because they're so small, they could become massive populations riding the coattails of these larger entities. And all I've described is an emergent process that, however much the details vary, is probably universal. Any planet that has mobile molecules and energy inputs can have this gradual emergence. The life forms that emerge may never get beyond single cells, but there will be this continuum of almost emergent life and emergent life. So everywhere there will be these small electron microscopic bits that can hitchhike along. Parasites are not simply universal, they're ubiquitous. And that's important because they are so small and because they don't have all this structure for metabolism and reproduction, they don't have to conserve their DNA like the emergent life. They have much more freedom to vary. In theory and empirically, these electron microscopic parasites or viruses will have much more diverse DNA sequences than life forms. Viruses are so diverse, in fact, that now when part of a sea or part of the Yangtze River is sampled and all the DNA is recorded, Virus DNA, which can be identified because of certain keys, is almost entirely different than any virus ever seen. All of it is entirely new. We've sampled such a small sample that we can't even estimate the diversity of virus, except that it's far greater than any life form. So bacteria and archaea, which I'll just keep them bacteria to keep it simple, inhabited this planet alone for 2 billion years, except they were endlessly attacked and are endlessly attacked by viruses. Half of all bacteria are probably killed by viruses. And every attack leaves a potential trace, a sequence left behind in a biofilm. Some bacteria survive with viral sequences left in their DNA from the attempted hijacking of their genome. Bacteria immune systems snip off DNA pieces from viruses and insert them in their own genomes for reference in case they're attacked again. This drip of virus sequences, they were not just any DNA, but far more diverse DNA than what bacteria had. So if most of it was damaging, it was also material that could be most useful. The nucleotides could be shifted, moved, and used. This drip continued billion year after billion year. If I have two glasses, one with fresh and one with salt water, and I take a dropper full of the salt and put it in the fresh, and then mix it and take the dropper full of the fresh out to keep it at the same level, and I do this for 8 billion years or even 100 times, the freshwater glass will soon have water just as salty as the saltwater glass. The steady drip of virus DNA, which was diverse, and thus sometimes useful, meant that, say, after 2 billion years, bacteria DNA was built from virus nucleotides. And since all complex life that's evolved after those 2 billion years, including us, is based on bacteria DNA, all of it is based on viral nucleotides. Our blueprints, our operating systems, our DNA is built from virus nucleotides. The emergence of life involves a continuum of potential, most unable to pass the threshold of self-replication. Many of these elements, though, parasitize emergent life, and they don't have diversity restrictions. They generate huge volumes of potential innovation and spread it widely, supporting genetic change. And this may occur on any planet where life emerges. Thank you.